continuation to the previous lectures, our next topic of isomerism is the stereoisomerism. Stereoisomerism is further has two types. Number one is cis-trans isomerism and second is optical isomerism. We will discuss cis-trans isomerism first. This type of isomerism is exhibited only by the compounds which contain double bonds. All those compounds which contain single or triple bond, they are not capable to exhibit cis-trans isomerism. Why it is so? It is so because whenever the organic compounds are synthesized, during synthesis, as a result of the carbon which contain double bonds, they will have two different groups. And if they have two different groups, during their synthesis, their groups attain different positions, and due to the pi bond in double bond that restrict the free rotations of the groups attached to the carbon atom, so they will attain a fixed position that will create a new type of isomerism which is called as a cis-trans isomerism. In case if the single bond is present, then all the group attached can rotate quite easily. If in case of triple bond is present, then only one group will left to attach with the carbon atom. That's why in the presence of triple bond, only linear geometry will develop, just like acetylene. But in case of all alkanes containing single bond, free rotation will take place, so no specific arrangement of the atoms or group of atoms in the space will be observed. But when there is a double bond, one is sigma and second is pi, then both of these bonds will stop the free rotation. The free rotation will not take place. Whenever the rotation will take place, overall whole molecule will rotate, but independently these will not rotate in this manner. That's why the pi bond restrict the free rotation of the groups and will create a cis-trans isomerism. Now, there are two basic assumptions or basic conditions for cis-trans isomerism. Number one, a double bond should be present between two carbon atoms, and secondly, all the groups attached to the carbon atom containing double bond must be different. If same type of group will be attached with the carbon containing double bond, then the question about the cis-trans isomerism will stop. For example, if I explain this CH2 double bond CH and then CH2 and CH3, this is one butene. Only these two carbon contain double bond. If these two carbon contain double bonds, these will be represented just like this. Depending upon the geometry, we can mention one hydrogen here, second hydrogen here, and this hydrogen might be represented here, or these groups are attached to this side. Now, as this carbon contains two same type of group hydrogen, that's why the cis-trans isomerism will not be exhibited by the one butene, because one side of the carbon contains same group, no doubt the other side of the carbon contains different group, but due to this carbon having same group, questions of cis-trans isomerism will vanish. But in case of 2-butene, if I mention this just like this, I will observe that both the carbon atom which are bonded with the double bond, they have different groups attached with them. For example, if I represent this is the double bond containing two carbon atom. Now there are hydrogen atom and methyl group, and also this contains hydrogen and methyl group. Now both of the carbon which are bonded with a double bond, they contain different groups and they adopt the position in space differently. Now, there is another chance. In this case, the cis-trans isomerism will be observed. It will show cis-trans isomerism. Now, if arrangement of these can also be just like this. Now, in cis-trans isomerism, we have to observe either the groups are present diagonally or not. If the groups are present diagonally, for example, methyl group is this and methyl group is this, in opposite direction, if the groups are present, then we will call this type of isomer as a trans isomer. But if the same type of group are present on same side of double bond and same type of group are present on same side of the double bond, then such type of isomer is called as cis isomer. Another point of view is, if we look along the side of the double bond, and if we observe that same type of groups are present on one side and the same type of group are present on the other side, then that will be definitely the cis isomer. And if we look along the double bond and we observe that different groups are present on both sides, then definitely that will be present as a trans isomer. This is the basic concepts involved in cis-trans isomerism. But remember, in cis-trans isomerism, we consider also the nature of atom or group of atom. For example, here in this case, for example, one carbon atom contain chlorine instead of hydrogen and other contain bromine. Then what will happen? That might be chlorine and bromine, say. But here that may be chlorine and bromine. Now, the nature of, no doubt these are the different groups, different atoms, but by nature these are called as halogens. And if I replace this methyl into ethyl, then that doesn't matter. It means that nature of this group and nature of this group is the same. Now, if we observe that the same nature functional group are present on one side and same nature group on the other side, then that will also give a cis isomer. But if these are present diagonally in opposite directions, then that will create a trans. This is a trans isomer, no doubt. Whatever the atom are, there might be same or different, but these are the different, but due to the nature present, these will be also called as a trans isomerism. And here, if I replace this into methyl, this will indicate that same nature atom or group of atom are on one side and same nature groups are on the other side. These will also represent the cis form. All these are points to explain how the cis-trans isomerism can be understood quite easily. Now, the next is optical isomerism. Now, next is optical. In case of optical, the necessary condition is all bond made by carbon should be single and one carbon atom should form 4 single covalent bonds. And the second point is all the groups attached to at the fourth position of carbon atom must be different. All the groups attached with the carbon atom must be different. Then definitely that carbon atom will show optical isomerism, and the optical isomerism showing center will be called as a chiral center, that will be represented by this. The simplest example to represent is, for example, see, in case of CH4, all the four valencies is occupied by the same type of atom. No question about the optical isomerism. Similarly, CH3Cl, no optical activity. CH2Cl2, no optical activity, no optical isomerism can take place. CHCl3, no optical isomerism. CCl4, no optical isomerism is exhibited by this. Now the optical, for example, if we take the example of amino acids, simplest amino acids is glycine. If we consider this amino acid, all the valency of this carbon atom, all the four valencies is occupied by the different groups. Now, due to all four different groups, this molecule can show optical isomerism. Definitely, in 3D structure, this molecule will be represented just like this. This is hydrogen, and this is NH2, and this is CH3, and this is COOH. Now, if we take its mirror image, that will represent just like this. Now, the mirror image of this compound cannot be superimposed on this original molecule, because in mirror image, with the all group which are on right will be shown in a mirror image on left. That's why such type of the molecule and its mirror image will not be superimposed on one another. That's why in such conditions we will call both of these as a optical isomer of each other. Such type of the isomers are called enantiomers, and in enantiomers the central carbon atom is represented with the star. This indicates a chiral center. This will be the chiral center, and depending upon the chiral center we will have optical isomerism. Each chiral center will develop two optical isomer. Now, optical isomerism can be judged, can be observed on the basis of this. Now, second, in such case, if all the atoms we use just like this, fluorine, chlorine, bromine and iodine. Now, in optical isomerism, the nature of the atom will play no role. It will not contribute. At all the four groups attached to carbon atom are different. Definitely, they will show optical isomerism because their mirror image will not be superimposed on the original molecule. That's why optical isomerism will be observed, and one chiral center create two optical isomers. There are so many questions present in the resource packs or past papers in which the students are asked to mention how many chiral centers are present in a molecule. Now, the students have to find out the carbon atom which are containing all four different groups. Now, if the molecules are larger, then keep on studying position with respect to their position, first position, second position, or along with the length of molecule. Whatsoever the length of the molecule is present, we have to consider all of them. Then we have to find out the point of difference where this can be observed that the carbon atom is containing different groups. So many examples are there in which cyclic structures are present, but we will continue to find out the point of difference on the basis of which we can mention the chiral center in the carbon. I have explained all the concepts regarding optical isomerism. Hope you have clearly understood all these concepts. Please comments, like, share and subscribe my YouTube channel for the upcoming video lectures. I thank you all.